Dear students, today we will talk about development of embryo in monocots. So embryo development is the predetermined, genetically programmed and metabolically regulated development of embryo from zygote. So how the zygote is formed? The zygote is formed by the fusion of the female gamete and the male gamete. Okay. So since already the embryo sac has the polarity, the chalazal and micropylar end, so same polarity is given to the zygote. This is zygote. The first transverse division occur in the zygote and it forms two cells. So the terminal cell and the basal cell. So basal cell is large, the terminal cell is small, and later on this terminal cell give rise to the proembryo and later on the embryo. So next division is the vertical division in the terminal portion and transfer division in the basal portion. So it give rise to the 2-cell stage proembryo. This whole is 8-cell stage, but if you see the embryo, this is 2-cell proembryo stage. Okay, and the lower cells, they form the suspensor cells. So they function to transfer the nutrient from to the embryo from the parent plant, or we can say from the endosperm. Then later on the transfer divisions occurs, and you can say the embryo from the 2-cell it forms 4 cells. So this is known as octant proembryo, and the whole is actually 8-cell stage. So 4 of them are suspensor cells. The last one enlarges and form the hostorial cells. This is also known as vascular cells, and its function is to absorb the nutrition and pass it to the suspensor cell, which later on passes to the proembryo. Okay, and then periclinal division occurs. So whenever there is periclinal division occurs, then outer and inner cell mass is formed. Transfer division keep occurring in these suspensor cells. So a stage comes called the globular stage, where you will see 3 different layers are formed. So here the differentiation occurs. So this is not anymore known as proembryo. This is a embryo stage, which is called globular embryo stage. And you will see the outer layer is called dermatogen, from which the epidermis form, the ground meristem from where the ground tissues develop, and then procambium from where the xylem and phloem tissue develops later on. Okay, and the first you can say this is the last cell of suspensor, this is the first cell of suspensor, and this give rise to the root cap, and last cell forms the hostorial or vascular cell. Then after number of differentiation occurs, it will assume this structure, and where it is also later on these will degenerate and this embryo is formed. So it will have one cotyledon in case of monocots, which is known as scutellum, and then it will have the embryonic axis, radicle and plumule. So we'll just see how it looks like. So when the ovule mature to seed, you will see this kind of a structure. So this is the whole ovule which get converted into seed. Now the endosperm, in case of monocot, they are endospermic seeds. So endosperm, large portion of the seed is covered by endosperm, and then you will see there is one cotyledon, and here you will see the embryonic axis, which bear at one end the plumule and another one the radicle, and there are coverings which is absent usually in case of dicots. So radicle has the sheath, protecting sheath, which is called coleorhiza, whereas plumule has protective sheath which is called coleoptile. Okay, and then same hypocotyle and epicotyle regions which are located in the seed. Okay, now let's quickly revise what is the difference between the monocot and dicot, you can say embryo. So monocot has just one cotyledon attached to the embryonic axis, and it is called scutellum, whereas there are two you can say cotyledons, and this one cotyledon occupy the terminal position, here you can see, whereas in dicot two cotyledons are there, but they are literal, side by side they are placed. Okay, so here the plumule is placed literal position, that is side. Okay, whereas in case of dicot it is near the origin, plumule is distal. And in monocot, a single cotyledon occupy terminal, we have already discussed. Coryptile and colioris are present in case of a monocot, where they are absent in case of dicot. So this is a brief story of development of embryo in monocots. Thank you.